Hello guys, uh, welcome to the next lecture in genomics, that is genome sequencing. We have in our syllabus, that is the genomics, we have seen the introduction previously, then we have seen a little bit applications about the genomics, then we'll see the third topic, that is whole genome sequencing, the methods, assembly and NGS platform. Okay, and then we'll see comparative genomics, structural genomics and functional genomics. Okay. So we'll see the genome sequencing, how and what it is. So in the genome sequencing, if you see that we are going to see in this lecture, that is the yeah, that is the what is mean by genome, then the types of different genomes, then what is genomics basically that we have seen already, but we'll see again how is genomics different from genetics, then the types of genomics, and then we'll start with our basic lecture, that is genome sequencing. Within that, we'll see the milestones in the genome sequencing, then technical foundations of genomics. That means what different techniques, what different methodologies we can use to sequence the whole genome. Then within that methodology, different steps are there. We are going to see that steps also. Then DNA sequencing approaches, then hierarchical shotgun sequencing, and within that what kind of markers we generally use for mapping and different things, then whole genome shotgun sequencing, and within that the new technologies which is coming up, genome sequencing achievements in different sectors that we are going to see, the benefits of genome research also we are going to see. Okay, so what is genome now? Genome is basically one complete set of genetic information, or the total amount of DNA content of that organism, or within the haploid set of chromosome. Now, guys, you all know that we have around 23 pairs of chromosome, right, within us. Out of that, one pair is sex chromosome, and other remainings are somatic pairs or the autosomes. Now, these up these pairs are just identical, of one copy just identical to other one. So if we say we need to sequence these chromosome, then we don't need to sequence the other chromosome, so we just need the haploid set of chromosome. Is it? We just need the haploid fragment or the haploid content of DNA, and that's what we called it as whole or total amount of DNA. This is very important. So we don't sequence the whole chromosome because it's a pair. Okay, and these complete haploid set is actually we called it as the genome. Not from the single chromosome, but from all the chromosome pairs, we need single haploid set and their sequences. The whole hereditary information of an organism is encoded in the DNA. In cytogenic genome means the single set of chromosome. Okay, so remember that term, cytogenic genome. Okay, it is denoted by X. The genome depends on the number of ploidy of organism. That means, suppose take example, there is a model organism called Drosophila melanogaster, or we can call it as fruit fly. Okay, so the diploid condition, that is 2x condition, and total around 8 chromosomes are there. Okay, and the pair, how many pairs are there? 4 pairs of chromosomes are there. So hexaploid is present in triticum aestivum. Okay, that means 2n condition. If you say the how many pairs, 7 pairs of chromosomes are there. Okay, and the 6 pairs are there, so there are about 42 hexaploidy are there. So the genome is found inside every cell. Okay, and those that have nucleus. So if you say talk in terms of suppose there is no nucleus, then where could we find the genomes? Take example of bacteria. Bacteria don't have a true nucleus, so that's why we can call it as prokaryote. And so the genome is actually present inside the cytoplasm. Okay, okay, the whole chromosome is present inside the cytoplasm, and so that's why we call them as prokaryotic. Okay, I guess you know that term. So a genome is situated inside the nucleus. Specifically, it is in the DNA, okay, in an organelle. So this is how exactly the DNA is present within the chromosome. If we take outside this whole nucleus, this is the cell, and if we take outside the chromosome, take example, we have 23 pairs of chromosome here, and if you take outside the single chromosome is actually looks like that. Okay, this is the one pair and this is the another pair. Okay, so if you take out the single chromosome out of it, and if we unwind it, these are histone proteins or histone complexes. Okay, which is actually important. These proteins are very important, where the DNA is engulfed around these histone complexes likewise, and it forms the coiled structure or super solenoid model. This is super solenoid model. This is solenoid models or this uncoiled structure. Okay, and this is how the DNA is actually looks like. So genes consist of DNA, which is made up of four chemical letters. That is adenine, is it, guanine, cytosine, and thymine. So out of that, these two are purines. Okay, and these two including one also uracil, these are pyrimidines. So guys, remember, in the DNA the thymine is there as a pyrimidine base, and in the RNA the uracil content is there instead of thymine. Okay, so remember that term. The term genome was introduced by H. Winkler in 1920, okay, to denote the complete set of chromosome and extrachromosomal genes present in an organism, including viruses also. How many types of genomes are there? Take example, prokaryotic genomes are there, eukaryotic genomes are there. So within that eukaryotic, there is a nuclear genome, mitochondrial genome, and chloroplast genome. Okay. So, if not specified, the genome usually refers to nuclear genome. Okay, if suppose in the genome, if you are looking towards the genome and it is not clear from which source the whole genome is there, whether it is mitochondrial or from its chloroplast, then we can say that the whole genome is from nuclear set. Okay, nuclear genome. Now, what is genomics? We have seen that in our introductory lecture, but we'll again repeat it. The genomics is the study of structure and function of whole genome. The genomics is comprehensive study of whole sets of genes. Okay, the genes which is present inside the genome. Their interaction rather than a single genes or protein. So comprehensively, how many genes interact towards each other in what way, that we generally study. According to T.H. Roderick, the genomics is the mapping and sequencing to analyze the structure and organization of genome. So the term genome was used by German botanist Hans Winker in 1920. Okay, the collection of genes in haploid set of chromosome is very important. Now it encompasses all the DNA in the cells. Genomics is the sub-discipline of molecular genetics devoted to the structure and function of entire genome, the mapping of genome. Okay, genomics contains these type of study actually, the sequencing of genome and analyzing the function of entire genome. Okay, the field includes studies of introgenomic phenomena such as heterosis, epistasis, pleiotropy, okay, and other interactions between loci and alleles that we have seen. The sequence information of genome will show us what they show: the position of every gene, the regulatory regions of genes, okay, which is present as a flanking region of each genes, regulatory regions, okay, promoters, operators, okay. Then the coding sequences that determines the proteins, okay. So triplet codon, we know the work of Nirenberg and Khurana. They have depicted or they decoded the whole genome within the 64 codon, and single codon governs a particular amino acid. Okay, we know the topic also, like degeneracy of code. Okay, so the Khurana and Nirenberg has actually give us that table for about 64 amino acids. Okay, sorry, 64 codons for 20 amino acids. How is genomics different from genetics? Of course, genetics looks at the single gene and genomics is looking at some big pictures from all the genes. Okay, so the types of genomics, there are three different types. The first is structural genomics. Structural means what? We determine the complete genome sequence of genome, and we construct the genetic map. Okay, and we actually say study the based on the map what genes is actually present in what different genome. That actually we do study, based on the genetic map. And there are like this has progressed in a steps as follows: the construction of high resolution genetics, and the physical maps are there, sequencing of genome, and the determination of complete set of protein in an organism. The next one is functional genomics. It refers to the study of functioning of genes and their regulations and the product. Okay, the functions. What different genes are there? What mRNA actually it producing? And after that, what kind of protein it actually expresses by translating? That actually we study, okay, based on the functional. And the other one is comparative genomics. It compares genes from different genomes to elucidate the functional and evolutionary relationship. So that actually different types of genomics. In the next lecture we'll see the genome sequencing. Okay, thank you very much.